Hey everyone, my name's Daniel and my presentation is on the neurotransmitter glutamate. So glutamate is also known as glutamic acid and it's an amino acid with a chemical formula of 5 carbon atoms, 9 hydrogen atoms, 1 nitrogen atom, and 4 oxygen atoms. It has a molar mass of approximately 147.13 grams per mole, and structurally it has an amino group, a central alpha carbon, a hydrogen atom, and two carboxyl groups. And this structure is polar covalent bonds with a polar anion molecule. It's highly water soluble, so it dissolves in water or dissipates or dissociates, and physically it's a white crystalline solid at room temperature and has a high melting point of around 247 degrees Celsius. Chemically, it's very reactive in amino acid metabolism and can be converted into other compounds. So glutamate plays several essential roles in the human body. It's produced by various cells, including excitatory neurons in the brain, astrocytes, liver cells, and kidney cells. And it acts as the main excitatory neurotransmitter in the CNS, where it supports learning, memory, cognition functions, and it's also involved in the amino acids metabolism and can be converted during the Krebs cycle or citric acid cycle for energy production. And these pictures shows the chemical formula of glutamate and the pathways in the brain that it goes through, like the striatum and the brainstem neurotransmitter centers. So glutamate is synthesized through multiple metabolic pathways, and one includes the enzyme glutaminase, which converts glutamine into glutamate through hydrolysis and exothermic reaction. So in the Krebs cycle, it involves glutamate dehydrogenase, the enzyme, and which catalyzes the reductive amination of alpha ketoglutarate to form glutamate. And overall, it basically means it breaks down and then does some decarboxylation, and it's exothermic, so it releases heat. And this is the pathway for that. Pretty complicated stuff, so I'm not going to talk too much about it. And this is the synthesis, so this is how it's made using NMDA, AMPA through the synaptic cleft. So, what happens if there's too much glutamate in your brain? So, it will affect your learning and memory. So, normally it's found in the synaptic cleft in concentrations of 1 to 10 nanomoles. And while there's high concentrations, it can cause a state of excitotoxicity. So this may happen with stroke, traumatic brain injury, or certain neurodegenerative diseases. And with excess glutamate firing on the receptors in the brain, it results in calcium influx in the neuron, oxidative stress, and neuronal death. So symptoms may include memory loss, cognitive impairment, seizures, and motor dysfunction. And it happens in diseases like Alzheimer's disease, Huntington disease, and amyotrophic lateral sclerosis. So what happens if there's too little in the brain? So too little glutamate is when the physiological range is less than 1 nanomoles in the synaptic cleft, which also disrupts brain function. And with a deficiency, it will diminish the excitatory neurotransmission, which leads to reduced communication, disrupted cognitive and motor functions. So symptoms may include feeling tired, memory and learning impairment, depression, as well as difficulty moving and coordinating. And it's found in diseases like schizophrenia and other behavioral mood disorders. So some treatments can involve using, well, first of all, you need a diagnosis. So you use clinical treatments in the lab, which involves neuroimaging and laboratory tests. So use an MRS, so magnetic resonance spectroscopy, to detect abnormal glutamate levels, or you can use a spinal tap, which involves tapping the cerebral spinal fluid and getting some out, which is a little bit invasive, and you can see the levels through this picture right here using the imaging. So diagnosis and treatment for it is using MNDA receptor antagonists like memantine, that's what they call it. It's a tablet and you just take it and swallow it, and it will eventually block the overstimulation by stopping receptor binding. So it's mematine is a fully synthetic compound created in laboratories, which selectively interferes with the receptors but allows the normal signals to go through, and it loosely binds, so it doesn't bind with all the glutamates, and it helps with Alzheimer's disease and calcium overload, and it will take several days to weeks to have the protective effects shown. So what happens if there's too little glutamate? You can treat that with D-cyclorylserine, which acts as a partial MNDA receptor and to promote presynaptic release of glutamate. So it can be found in natural antibiotics produced in soil bacteria, but it's also pretty much just synthetically made and administered orally. And the therapeutic effects for it is which it protects neurons within days to weeks and for longer term improvements in cognitive mood. So these treatments restore the balance of excitatory neurotransmission and prevent further neuronal damage. And these are my references. Thank you for watching.